Hello, good evening. Welcome. Wow, I think this may be our best crowd yet. Not entirely surprising given that it only took 14 hours for this event to sell out, which is definitely broke all records. I will blame Tyler. So my name is Anna de Konnik. I'm the Executive Director of the Koch Institute. And as many of you know, we are celebrating our 5th anniversary as the Koch Institute in this building. And for the occasion, we changed our regular standard solutions with Insight format and decided to have 5 masterclasses, one for each of our 5 focus areas. And so we've already covered metastasis and devices for cancer detection and monitoring. In the fall, we will have nanomedicine and personalized medicine. And tonight, this evening, our focus will be on immuno-oncology. I want to thank the Koch Institute postdocs, grad students, and staff who were in the galleries volunteering for our showcase this evening. And now it's my pleasure to introduce Koch Institute Director and David H. Koch Professor of Biology, Tyler Jacks. Before I give him the microphone, don't get ahead of yourselves. I just want to say brief introduction of him. He received his bachelor's at that other Cambridge University, if you can believe it. He went and did his PhD with Harold Varmus, who was then at UCSF, and then he did his postdoctoral studies with Bob Weinberg at the Whitehead Institute. He was recruited to MIT as an assistant professor nearly 25 years ago, I know. Unbelievable. And he became director of MIT Center for Cancer Research nearly 15 years ago. And I have had the honor and privilege of working with him for 13 of those years. Tyler is a Howard Hughes investigator, a member of the National Academy of Sciences and the American Academy of Arts and Sciences, of which the president is with us tonight. So that's very exciting. He and his lab have published over 200 scientific peer-reviewed publications. He has received numerous awards, including the Killian Award, which is the highest honor that MIT faculty can bestow on one of their colleagues. And he sits on numerous boards of directors, scientific advisors. He's a past president of the American Association for Cancer Research, the current chair of the National Cancer Advisory Board, and was recently named a co-chair for the White House Cancer Moonshot Task Force's Blue Ribbon Panel. That was long. Please join me in welcoming Tyler Jacks.
Well, thanks very much, Anna, and thank you all for coming out tonight. This is a wonderful turnout. Anna described it as the best crowd ever, certainly the largest crowd ever. We'll wait and see if you're the best crowd ever. So what I'm going to do over the next maybe 35 to 40 minutes is tell you about this field of immuno-oncology. And I'll end by talking to you about some of the efforts taking place here in the Koch Institute and specifically in my lab in that area. So we'll start general, and then we will narrow down to some specific and mostly unpublished observations that we've made most recently in this really exciting area. Now, this is a bit of a daunting task, I must say, firstly because this is a master class, and I realized after I accepted the invitation to give this, I had no idea what a master class was. So I believe that I am supposed to educate you in an important area of science. And this is a diverse audience that we have with us. We have alumni who have little background in the field, if any. We have industry executives who work directly within this area. We have the former president of MIT, who herself was a life scientist before she became president, Susan Hockfield. And we have the chairman of the corporation of MIT, Bob Millard, who knows nothing about biology, though he did take Eric Lander's course online, so he has that going for him. So I'm going to do my best to explain this topic, which is really fabulously exciting, in a fashion which will be accessible to those who didn't have a background, and hopefully still interesting to those who did. So let's get started. This area of immuno-oncology, which has taken the field of cancer research and cancer treatment by storm over the last few years, is not new. This is Paul Ehrlich, who is a German physician scientist who practiced and did research in the era between about 1870 and 1910. He actually won the Nobel Prize in 1908 for his important discoveries related to immunology. And Paul Ehrlich, in around 1900, suggested that the body should have the ability to recognize cancer as being different from normal cells, and thereby limit the development of cancer in human beings. And moreover, that we might be able to use the immune system to fight cancer, to treat cancer, and maybe to prevent cancer. Now, this was a prophetic statement, a series of statements, and in essence, Paul Ehrlich was right. But it took a really long time for his predictions about the use of the immune system in the treatment and control of cancer to come true. But now, in the year 2016, we've seen that, in fact, through a long process of basic science and clinical development in academia and industry and government labs, we in fact have the tools to use the immune system very effectively to treat cancer. In 2013, Science Magazine described cancer immunotherapy as the breakthrough of that year. This was based on the science that had been done that taught us how cancers avoid detection by the immune system and grow despite the presence of cancer cells, immune cells to fight cancer cells, and how we might be able to stimulate those cancer cells to fight back. And in this year, just earlier this year, the American Society for Clinical Oncology named cancer immunotherapy the clinical advance of the year, based on the kinds of clinical experiences that are being seen today using the immune system to fight cancer more effectively. The interest in this field is evidenced by the very large number of pharmaceutical companies and biotechnology companies that are now investing in this area. This is actually a partial list. The actual number of companies that are working in this area is probably at least twice this large. You can see virtually all the major biopharmaceutical companies are working in the area of immuno-oncology today, and they're investing heavily. Most oncology programs in these companies are geared to figuring out how to use the immune system more effectively to treat cancer. And every day we read about another one, and that's not an overstatement. Just today in the Boston Globe was reported that Agios, a company not far from here that works in the area of cancer metabolism, has decided to work in the area of cancer immuno-oncology because it's so hot and so powerful. And here they're describing a deal that they signed with Celgene to pursue their technologies to help us understand how to use the immune system more effectively. And we here in the Koch Institute, where we're very interested in moving our ideas out of the lab and into the marketplace where they can be developed more successfully, we too have started a number of companies in this area of immuno-oncology, and those are shown here. These are started by several of my colleagues, both within this building and also affiliate members of the Koch Institute, and I'll call special attention to Equipoise Therapeutics, which is a company that we've recently started, my lab, in collaboration with David Rollet at the University of California, Berkeley, and a co-founder, Bill Haney, who happens to be sitting right here in the front row, to try to use another cell of the immune system to effectively treat cancer. And I'll describe briefly work that we're doing in that area. So many of us are engaged in the laboratory as well as in industrial settings to try to take advantage. And these aren't just ideas. This is not just the promise of using the immune system to more effectively treat cancer. There are, in fact, new drugs, FDA-approved drugs, that are doing exactly that. There are now 4 approved drugs in a category we call immune checkpoint therapies. 3 of the 4 are shown here. The first of these was called Yervoy, developed by Bristol Myers Squibb. And I'll describe to you what Yervoy does. But it was the first example of one of these immunostimulatory drugs which was having a huge effect, initially in melanoma and then in other cancers. And 2 other drugs which target a different protein, one again by Bristol Myers Squibb, one by Merck. And you can see that these drugs approved quite recently, 2011, 2014, are already being sold widely and benefiting patients significantly. So this is not just promise. These drugs are having an impact in the clinic, in patients, and in companies. So those are some of the numbers, but I think the power of the story is best told through the personal experiences, like this individual here, whom I hope is recognizable to you. This is former President Jimmy Carter, 90-year-old man. Why is he smiling? He's smiling because he's just told that he's cancer-free, which is quite remarkable because he had relapsed malignant melanoma, which had metastasized to his brain. 5 years ago, that was a death sentence. Nobody came back from that diagnosis. Jimmy Carter was treated with one of those immune checkpoint drugs that I just showed you on the previous slide. He is now cancer-free and likely cured. Another example. This is Emma Whitehead. You can see her here when she was relapsing from acute lymphoblastic leukemia, which she had been diagnosed with when she was 5 years old. She was treated with standard treatments. They failed and failed again. She and her family were out of options. Fortunately, researchers at the University of Pennsylvania had developed a new immune-based therapy for this type of cancer, and they were testing it on adult patients. Her family found out, contacted the doctors. They were willing to test to treat her. And this is Emma today, cancer-free. And last year, she had a very special visit with this President of the United States. And if you can read this, it says, "Please excuse Emily from school. She was with me." So 2 examples, a 90-year-old individual, a 7-year-old individual, benefiting from these treatments that mobilize the immune system to more effectively eliminate cancer cells. 2 examples. Sometimes we worry in science that anecdotal examples are not necessarily representative of the experience of a much larger number of patients. But in fact, as we look at more and more patients being treated by these immune stimulatory agents, we find that, in fact, it's working quite well. This is what we typically see in cancer clinical trials. And I have to say that curves like this are incredibly disappointing and disheartening to me. This is a schematic view of one hypothetical clinical trial where individuals are treated with a standard agent and nobody survives after a period of 1 to 2 years. And here's the new treatment being developed, and the patients live a bit longer, but really not much longer. And at the end of a period of time, no one survives. This would be an FDA-approved drug based on that very short window of improved survival, because that's the best we could do. But in fact, the new treatments, these checkpoint therapies and other immune-based therapies, are showing curves that look like this, where a significant number of patients are responding. Here, about 20-25% of patients are responding, and they're not responding and then relapsing a few months later. They're responding, and their disease goes away and seems to stay away for years. They may be cured, or their disease is controlled for the long term. This is a schematic view, but the data actually look like this. So here is an example from malignant melanoma. This was a study published 5 years ago with what was then a very promising new targeted treatment for melanoma based on a known mutation that takes place in a pretty high percentage of melanomas. It's a mutation in a gene called BRAF, and companies made drugs that inhibit the function of that mutant protein with the hope that that drug would be a precision treatment for that form of melanoma. Here's what the control patients do with standard chemotherapy. They don't do very well. They only live for a certain number of months, and at the end of that period, only 8% or so of the patients are alive. With this new targeted treatment, there is an improvement. You can see it here. If you look at the 50% survival rate, these patients might live 3 or 4 months longer on average. But note that everybody relapses, and the overall survival rate is not much better. Nevertheless, this drug was heralded as a major breakthrough for the treatment of melanoma and approved with great fanfare by the FDA. By contrast, look at what we're seeing with respect to immune-based therapies for the same disease. Here's standard of care treatment. Nobody does very well. Here's one of these immune checkpoint inhibitors. In this particular case, the drug by Bristol Myers Squibb called Opdivo, which blocks a protein called PD-1, 40% of patients are surviving and surviving long term. We now know they're living out 2 years, 3 years, 5 years, perhaps 10 or more years. Remarkable change, remarkable improvement. And this is happening fast and furiously. This is a timeline that shows the FDA approval of several of these so-called checkpoint inhibitors. The first of these I mentioned was Yervoy by Bristol Myers Squibb. Merck had Keytruda, which blocks PD-1, approved shortly thereafter. BMS again, their PD-1 inhibitor. All three for melanoma, note. And then certain other cancers, lung cancers, renal cell cancers, bladder cancer, Hodgkin's lymphoma, all showing sensitivity with similar kinds of response curves that I just showed you for these new agents. Dramatically different, like nothing we've seen before in the treatment of highly aggressive cancers. You can see why there's such optimism in this field. So you're probably thinking to yourself, well, that looks quite good. These single agents are having dramatic response rates, 20% of patients responding, 40% of patients responding. If those individual drugs work so well, perhaps there's an opportunity to do even better by combining them. In fact, that's been tried. If you add 2 agents together, you might be able to boost the response rate from 20 or 40% to an even higher number. I can tell you that combination clinical trials with these drugs are ongoing across the country and throughout the world. There are hundreds of clinical trials ongoing today with individual checkpoint inhibitors and other immune-based therapies for cancer and combinations thereof. And some of these are showing quite dramatic effects. We're going to focus again on melanoma. Here's one drug, and I showed you data similar to this before. This is the anti-CTLA-4 drug, Yervoy. It works in about 25% of patients. You now combine it with the PD-1 acting drug. The combination is giving response rates of nearly 60%. Those of you in the audience who are a little further along in life will remember that malignant melanoma was a death sentence. It was like glioblastoma. It was like pancreas cancer. These patients, 60% are surviving. It's a completely different story now. And I suspect that this is just the beginning. There will be many more cancers like malignant melanoma, and we have data to support that statement in the years to come. So that's why we're so excited, because these drugs are working in ways like we've never seen before. So now I want to teach you why they work. This is the Masterclass part. So to understand how these drugs work, why they work, you need to know a little bit about your immune system. So the essence of your immune system is to distinguish self from non-self. So what do I mean by that? Self is you. Non-self is stuff that invades you, like bacteria or viruses. Your immune system is built to recognize those foreign invaders and to eliminate them, to block their activity or to kill them. Your immune system is designed to recognize things that are specific to them, that are therefore different from you, and to get rid of those things that are different. And the immune system has important qualities that allow it to carry out that function. Selectivity. Cells of the immune system evolve in your body to be selective to those things that are different from you, selective for a bacterium, selective for a particular virus. And it's adaptable, so that even if the bacterium or the virus were to change over time, your immune system can change with it. And you can probably see already why that's a good thing when we think about cancer, because cancers change also over time. And if the immune system can change with them, the immune system can stay active. And there's another benefit as well. The immune system has something we call immunological memory. And you know this is true because all of you have had vaccines. And the reason that you've had vaccines is to train your immune system to having been exposed to some part of a pathogen, a bacterium or a virus. That's what vaccines are. They're a part of one of those things. Your immune system thinks it's seen that thing, and it establishes this immunological memory where cells are actually set aside in your body in case that you get infected by that thing in the future. And if you do, those cells become activated quickly and eliminate that pathogen before it causes symptoms in you. That's the way vaccines work. Another very powerful feature of the immune system that we'll take advantage of. So with this in mind, we have to ask the question, where do cancer cells fit in? Are they self or non-self? They come from you. Cancer cells arise from normal cells in your body. So in that respect, they're self. But they actually change over time as they develop from normalcy to malignancy. And in that respect, they're non-self. So which is it? Well, in fact, since Paul Ehrlich made his claims about cancer immunology way back when, we've learned a tremendous amount about how cancer cells develop in the body. We knew nothing about it at his time. We've learned a hell of a lot about it over the last century plus. And it boils down to the fact that cancer cells arise from normal cells through the acquisition of mutations, changes in your DNA, which change the function of genes and change the function of proteins. Some of those genes are called oncogenes, others called tumor suppressor genes, completely irrelevant for tonight's lecture. Just know that cancer cells carry altered versions of cellular genes. And those genes become altered because over your lifetime, your DNA gets damaged. Sometimes you inflict it on yourself. If you smoke, you're causing damage to your DNA and mutations. If you expose yourself to UV light, you're causing DNA damage and mutations. That's why those two things are carcinogenic. They accelerate mutation, they accelerate cancer. Some of it is due to exposures like this, but I have to tell you, a big part of it is due to random chance. Your cells aren't perfect. They make mistakes. They acquire changes as we age. So some of that is unavoidable, in fact. In addition to the changes that are taking place within these cancer drivers, there's a lot of collateral damage as well. The DNA of the cancer cell is being showered with mutations. It's acquiring errors all over the place, which is causing alterations that the immune system can also see. In aggregate, these altered genes provide the targets that differentiate cancer cells from normal cells for the immune system to recognize. Non-self from self. I also wanted to mention that sometimes, but not frequently, cancers arise due to viral agents. The classic example is cervical cancer, which arises due to infection with human papillomavirus. That's yet another set of antigens for the immune system to see. And there are vaccines that work to prevent cervical cancer by eliminating the viruses that cause that disease. We're not going to talk much about prevention using the immune system in that way, but it's important for you to know about it. In addition to the changes in gene structure and altered proteins that I've emphasized here, I also wanted to mention that cancer cells become abnormal in other ways. They change the expression, the activity state of genes, even if not mutated. They'll turn things on they shouldn't turn on. They'll turn on proteins that are normally only expressed in the embryo, but begin to express them in the cancer cell. And those too can be seen by the immune system as abnormal and foreign. Okay, so what does the seeing? Well, we're not going to dwell on this because I could give you a semester's worth of information about how the immune system really works. So I have just one slide to introduce you to the many cells in your body that are dedicated to finding foreign things and eliminating them. Many different cells. We're going to focus on just one of those cells today. This cell here, I'll just mention in passing B cells, which are antibody-producing cells, very important in this whole story. And I'll come back towards the end to talk to you about natural killer cells, which have gotten less attention in the fight against cancer, but are gaining interest, and that's what our new company is focused on. But really, most of the action is here. These CD8 positive cytotoxic T cells. These are natural-born killers. These are the trained killers, the junkyard dogs of the immune system. And here's one T cell at work. This is an actual photomicrograph of a cancer cell interacting with a cytotoxic T cell. And this kiss of death is a literal thing. When that interaction takes place, the T cell gets activated, secretes factors, and kills its target. And that's exactly what you want to have happen for a virus-infected cell or for a cancer cell. You want those T cells to get engaged and kill the target. And if that can work, you can eliminate cancer cells and keep them gone. It turns out that for most cancer patients, you have cells like this that can recognize your cancer. As your cancer cells evolve from normal cells and acquire those mutations, your immune system recognizes those changes, and you make cells like this, which almost certainly in the early stages of your disease are fighting the cancer, trying to fight the cancer. The problem is it doesn't last, at least for many patients. Those who develop more advanced cancers, it doesn't last. The cancer fights back. The immune system turns itself down, desensitizes itself. And we're beginning to learn the mechanisms by which that inhibition of the immune response occurs. And these are the targets of the checkpoint inhibitors. So here's an example of one. Here's a cancer cell which is engaged with a T cell. Now this would normally activate the T cell to kill its target, the cancer cell. But the cancer cell has figured out a trick. It's turned on this protein here called PD-L1, which is engaged with a protein on the surface of the T cell, and that sends a negative signal to the T cell which shuts it off. And it becomes a non-killer. This is a T cell which is called in the field exhausted. It's there, but it's not capable of being activated to kill its target. However, knowing this now, we could make drugs which block this interaction, antibody-based drugs which block PD-1 or block the protein on the cancer cell, PD-L1, block that engagement from happening, and in that way activate the immune system to kill again. And that's how the checkpoint inhibitors work. They re-engage exhausted T cells to function to kill their targets. Now, I've mentioned already two of the approved drugs in this class. CTLA-4, which was the first discovered by Jim Allison, first at Berkeley, then at Sloan Kettering, now at MD Anderson. He will win the Nobel Prize. You can bet on it. PD-1, I've mentioned. Major work done by Arlene Sharp, Gordon Freeman, Harvard Medical School. The ligand for PD-1, PD-L1, also approved. But you can see that there's a whole bunch more targets on T cells that can either inhibit their function, such that we want to make inhibitors of the inhibitors, or proteins that we know that if we were to activate them would make T cells function better. And all of these and more are being developed by drug companies to enhance immune responses against cancer. That's why all those companies on that earlier slide are so busy at work. Okay, so I've told you that you've got T cells in your body that can recognize and eliminate cancer cells. I've told you that some patients with cancer, malignant melanoma, non-small cell lung cancer, bladder cancer, renal cell cancer, are responsive to these checkpoint inhibitors. But what I haven't told you is that not all cancers are. Certain forms of breast cancer, it seems, are not responsive to these drugs. Prostate cancer, in some forms, seem not to be responsive to these drugs. So a major question in the field is why? What distinguishes the cancer types and the cancer patients who respond from those who fail to respond? I've given you a major clue as to why that is. It took scientists and hundreds of thousands of dollars of the NIH's money to solve this problem, but you sitting in this room should know the answer to this question. So I'm going to ask you. If you came into the room knowing the answer to this question, please don't answer. But if you didn't, suggest a reason why malignant melanoma might be very responsive to these drugs. Lung cancer might be very responsive to these drugs. Why do you think? Don't make me call on you. So one possibility is they don't turn on these inhibitory receptors. Perfectly good possibility and probably right at some level. Maybe there's not enough immune cells. Another good suggestion. But I want to focus on those who respond. Why do the responders respond? Say it again. Well, they are caused by the basic genetics, but I emphasized already that malignant melanoma, it's UV-induced. Lung cancer, it's smoking-induced. These are cancers that are showered by carcinogens. Their genomes are way screwed up. They respond because there's too much collateral damage. There's too much collateral damage. And we now know this by studies that have looked at the genomes of all sorts of cancers, which are spread out across the top here. And this graph illustrates the mutation frequency in those cancer types. The ones further to the right have more mutations. The ones to the left, fewer mutations. The cancer types that respond are over here because the immune system has lots of things to see in them. They've got lots of mutations. They look very different from normal cells. So there's lots of T cells that can see those cancers. Whereas for other cancers, there are fewer mutations. They look more normal. There are less things for the immune system to see. So that's a major differentiator between the cancer types that respond and those that fail to respond. It's not perfect. Expression of the receptors is part of it. Numbers of T cells is part of it too. Some of these patients who have a high mutational load don't respond. We need to figure that out. Some of these patients who have a low mutational load do respond. So we have to figure that out. But mutational load, the number of mutations present, seems to be a major differentiator between those that respond and those that do not. Okay, sticking with this slide for a second, and given what I just said, you might worry about patients over here because they don't have much in terms of new mutations for their immune systems to see. And I want to highlight one, this one, ALL. That's the disease that Emily Whitehead had. Very few mutations. Her cancers probably had 5 mutations that differentiated normal blood cells from her cancer blood cells. Very low likelihood that the immune system would see those cancer cells naturally. So if the immune system can't see the cancer cells naturally, if you didn't evolve T cells in her body to see those cancer cells, what could we do about it? It's another question. Anybody want to hazard a guess? We still want to use the immune system. It's so powerful. And I actually showed you that it cured her. But we're not going to rely on the natural immune response to her tumors because it's probably not good enough. You could think about vaccinating, and if you had a good antigen to vaccinate with, that would be a good idea. But what if we took her T cells out of her body and engineered them to see her cancer? Maybe that would work. And that's exactly what Carl June did. Based on earlier work from Steve Rosenberg at the National Cancer Institute, who showed that you could take T cells out of a cancer patient, grow them up in the lab, and sometimes observe good responses when you return them to the patient. Steve Rosenberg and now scientists at Juno Pharmaceuticals and Kite Pharmaceuticals are going one step further. They're taking T cells out of a patient, growing them up in the laboratory to very large numbers, and then using genetic engineering to engineer those T cells to see that patient's cancer, to express on the surface of those T cells a receptor that can recognize those cancer cells, engage with those cancer cells, and kill them. Not based on a mutation present in those cancer cells, but just based on something that's expressed on the surface of those cancer cells. These are called chimeric antigen receptor T cells, and they work quite well. More than that, I would say. Here's the clinical study published by Carl June's group a few years ago. They treated 30 patients, adults and kids, with ALL, and they observed at 6 months, and this seems to be lasting out for most patients, an almost 80% response rate. These are heavily pretreated individuals who had relapsed refractory ALL. Without this treatment, they would have died. 80% likely cured. So engineering T cells to recognize cancer cells can also work. This is a promising therapy, not yet approved by the FDA, but it does have one limitation. You have to take the cells out of the patient, grow them up to large numbers, engineer them in the laboratory, and then put them back in. It's complicated, it's expensive, and companies are hard at work developing them. One example shown here, an FDA-approved product from Amgen, and for the purposes of full disclosure, I'm on the board of directors of Amgen. But this is a drug called Blincyto, which more or less does exactly what I said that CAR T cells do, but instead of engineering T cells that you take out of a patient, you give the patient a protein. It's like a matchmaker, a protein that you've engineered that can bind to T cells on the one side and cancer cells on the other. It links the two together, and that causes the T cell to kill its target. And this has been approved, actually, for the same disease that Emily Whitehead had. And I was actually just out at Amgen at a board meeting, at the annual meeting of stockholders, and the CEO described the approval of this drug. And one of the stockholders raised his hand and said, "I just want to thank you. I have a grandson who was diagnosed with this disease, and his doctors had given up, and you cured him." Another drug, or another strategy, which we won't spend too much time on, sadly, because it's very promising, involves vaccines, and that was mentioned by somebody, including vaccines based on using viruses that you can inject into the tumor. They cause cancer cells to die. They cause the proteins that are present in the cancer cells to be presented to the immune system, to charge up the T cells and kill the cancer cells. These oncolytic viruses have been approved for melanoma treatment, and other ones are being developed for others. You might have heard about the use of recombinant polio viruses for the treatment of glioblastoma. This is the way they work. They kill the cells in the brain and activate the immune system to do further killing. Other vaccine strategies are also really exciting, and you'll hear more and more about those as the field matures. Okay, so now I want to change gears and finish, hopefully in about 7 minutes, with some stories from my laboratory. Anna mentioned that we're celebrating our 5th anniversary and highlighting the various research areas that take place within the Koch. And tonight we're talking about cancer, immunology, and immunotherapy. Many of my colleagues here work in this very exciting area. Some of them, for example, are developing fantastically new and exciting vaccine strategies for cancer that stimulate much better immune responses against the disease. Others are designing fantastic new molecules that stimulate the immune system in new ways. In my lab, we too have been focused on the immune system. Starting about 10 years ago, we decided to use our expertise in developing mouse models of cancer to see if we can understand how the immune system interacts with the disease in ways that hadn't been possible by studying human patients. Because in human patients, you have limited access to material, But in the mouse models of the disease, you can manipulate things very carefully. And we can learn much more through this process. So we've been using these genetically engineered mouse models for many different purposes. But for tonight, we're going to focus on using these models to study how the immune system sees cancer. Again, more detail here than you need to know. Suffice it to say that we can induce cancers in mice using genetic tricks. We can pull levers on the genes of the mice and introduce mutations, thereby causing cancer cells to arise. And we can engineer those cancer cells almost in unlimited ways. And in this particular application, we've engineered the cells to express something for the immune system to see, an antigen that T cells can see. And when we do that, we can in fact invoke an immune response against the cancer. This is a tumor in these mice. It's a tumor in the lung. And these brown cells that you see here are immune cells invading into the tumor and destroying those cancer cells. That's a good thing. In this mouse model, we can observe T cells interacting with the tumor and causing damage. And I say that because we can observe that these antigen-expressing tumors shown in black don't grow as well as non-antigen-expressing tumors shown in white. This difference here is a function of the immune response to the cancer. But you'll also notice that's true at 16 weeks after the initiation of these tumors. But notice at 24 weeks, the effect goes away. The immune system seems not so capable of controlling the tumor at that point. And the reason is that at that point, the T cells have left the yard. They're nowhere to be seen. They're actually sitting on the sidelines, literally. They're sitting on the edge of the tumor, but they're not inside. They're not doing their job. In fact, they're exhausted. If we look at these T cells, for these various factors, which we know can control immune function, and I've mentioned CTLA4 and PD-1, and I'll mention some others in a moment. In fact, they have markers of exhaustion. This is a graph that shows that at that late time point, the levels of this checkpoint protein PD-1 are high in those cells, correlating with their lack of function. So you might imagine to yourself, if I could reverse that, if I used an anti-PD-1 antibody from my friend Arlene Sharp, I might be able to activate those T cells and get them to kill. Well, we did that. We got some antibodies from Arlene Sharp, and God damn it, they didn't work. Control tumors grow in black. PD-1 treated tumors grow in red. In this particular tumor, PD-1 is ineffective. It's there, probably doing something, but it's not controlling the activity state of those cells. Something to learn there, something to study. But there are lots of other things that control immune responses, including these other checkpoint factors, but also cells. Cells in the microenvironment of the tumor that themselves control the activity state of T cells, including this very important cell type here called regulatory T cells. You can tell from the name that they function to regulate the activity of other cells. And literally, they function to inhibit the activity of those T cells. They exist in your body to control autoimmunity. If you don't have regulatory T cells, if you're a mouse or a human, you have raging autoimmune disease. So you need these regulatory T cells to dampen down your immune response against self. So we wondered, might the regulatory T cells be involved in this process? And so Nick Joshi in the lab looked, and he found that indeed at the late time points when the immune system was suppressed, levels of these regulatory T cells within the tumors was increased dramatically. So they're there, but this is guilt by association. Are they actually functioning to control the immune response? So third question and last, if you wanted to test whether the regulatory T cells were controlling the immune response in this cancer, what would you do? Get rid of them. Get rid of them. And fortunately, because we're working in the mouse, we can do virtually anything we want. So we pull out a few other genetic tricks and allow us to deplete the regulatory T cells. After the tumors have evolved, we get rid of them. So what happens? Well, this is the before picture. Lighting isn't great here, but this is a tumor. The tumor cells are stained in red. This is a healthy, growing tumor. Bad news for the mouse. Here are the T cells in green, hanging out on the sidelines, not doing their job. Regulatory T cells surrounding them. We now deplete the regulatory T cells, and look what happens. T cells get hyperactivated, they proliferate, and they charge into the tumor, and they cause massive tissue destruction. So this is evidence that in this model system, regulatory T cells are controlling immune responses, and therefore if we could figure out ways to eliminate or inhibit regulatory T cells in cancer, we predict this would have a major effect. So we're trying to figure that out, and many companies are likewise trying to figure that out. Lastly, I wanted to mention, or almost lastly, I wanted to mention this other cell type that we've turned our attention to more recently, and those are the NK cells. These cells, discovered in the mid-1970s, are known to be able to attack and control cancers, but their ability to do so is probably limited by their ability to get access to the cells in an established tumor. So Leah Schmidt in the lab took a page out of the former student's book, Michael Dupage, and she built a vector that allows us to engineer those lung cancers to express not just an antigen that the immune system, that the T cells can see, but also something that would attract NK cells to the tumor. And she did the following experiment. She introduced cancers in these mice, or induced them, in a fashion where they express just the T cell antigens. And I've shown you what happens there. Tumors are inhibited, but they continue to expand at later times. And then she combined the T cell antigen with ligands to attract these natural killer cells to see whether there would be an enhanced effect through the addition of NK cell infiltration. And indeed, what she found was that in contrast to this T cell antigen only expressing tumors, which grew relatively large, and there were a large number of them, if you express the combination of NK cell ligands and T cell antigens, the number of tumors and the size of those tumors was inhibited. So NK cells can, in fact, augment the activity of T cells and probably do some killing on their own, and in this way provide another lever to control established cancers. And so therefore we believe, based on these and other data, that in addition to checkpoints and other ways to stimulate T cells, that NK cells can provide another tool to enhance anti-tumor immune responses. And it's based on that we started this company to explore ways to do what we've done in the mouse in a human being. So lastly, and then I'll entertain questions, I've been focusing on our use of these model systems in the context of therapy. How can we use the immune system to treat cancer once it's already established? I'm also super excited about the prospects of using the immune system to prevent cancers from forming in the first place. So can we use the immune system to train it to see something that the cancers will eventually express in the same way that you do to prevent a viral infection in the context of vaccines? And in fact, in human cancers and in these mouse cancers, we know of targets like this one here that get upregulated in the late stages of cancer. This brown staining shows these proteins present in the late stages of cancer. They're not present in the early stages of cancer, and in fact, they're not present in the adult animal. This is an example of what I told you before. This is a protein that's normally restricted to embryogenesis, but it gets abnormally activated in cancer. So given that we know that that's true, could we develop a vaccine against this target and prevent cancers from forming in the first place or progressing? And so we're doing this experiment. David Kanner in my lab, in collaboration with Omar Khan and Jasdev Chahal from the Anderson and Langer labs in the building, are developing a vaccine against this protein, vaccinating the mice that will one day get cancer using a very fancy nanoparticle-based vaccine strategy, which is super effective, and then inducing cancers in these mice to ask the question, will it prevent the development or the progression of the disease? This is an ongoing experiment. The early preliminary experiments look really encouraging, so I'm quite optimistic that we can use the immune system in this way as well, not just to treat cancers, to prevent them from occurring. And so I'm going to stop there and remind you that cancer research and progress against cancer sometimes takes a long time. But fortunately, through the sustained efforts of researchers across the world, and including here at MIT, we've made great progress. And I'm extremely optimistic that using the tools of the immune system, we will one day be able to control most cancers and prevent many more. Thanks very much.
Thank you, Tyler. I think we now know why this was called a masterclass, because that was masterful. Questions?
Yeah.
In the therapy, the ex vivo T-cell activation therapy, when you're dealing with a cancer cell that is of low antigenicity, that is not much foreign,
Yes.
how do you generate or screen for the appropriate cancer-specific or cancer-binding
Yes.
moiety?
Very good question. And in the specific example that I gave you, treatment of ALL, they actually didn't rely on something that was specific to that individual's cancer. They simply targeted a protein that's present on the surface of all B cells and all cancer cells that look like B cells. The treatment gets rid of all B cells and all cancer cells. So it's a little bit like carpet bombing in that particular case, but you don't need B cells, it turns out. You can get by without them. In other cases, you would have to study the genomes of the cancer cells to find what's specific to them, and then engineer the receptor to bind to that specific thing. Yeah.
Wonderful lecture, thank you. How is your research coming towards SRCC, which is a growing segment, and the tumors are not being treated the way with what all the agents we have out there? What's going on in that field in your world?
SRCC.
Yeah. The signet ring cells are?
So
Thank you.
we are not working directly on it, and therefore I don't know any specific details. It is a tumor that I think is undergoing genomic analysis, and one thing that I failed to mention, but should have, is that as we learn the detailed genome sequences of any given cancer, it raises the possibility that you can develop vaccines that are specific to the changes within that particular cancer. And that's an approach which will cut across all cancer types. Very personalized, individualized treatment. So that's as much as I can say. Whether there is additional work using the immune system for that particular cancer type, I just would not know. In the back, yeah. What are we doing with them? We are attempting to stimulate their activity and recruit them to tumors based on the receptors that we know that they express and targets on cancer cells that we know are present. So it's a strategy to attract NK cells and activate them, to mobilize them and activate them and keep them from being inhibited. So it's a combination of those three things that we think will reproduce what we've successfully done in the mouse in the context of a patient. Yeah.
What about T-cell ALL?
T-cell ALL. So again, we don't work on it in my lab. The good news is that for all leukemia types, the strategies that utilize either CAR-T or BiTE-like, these are the proteins, are particularly effective. So leukemias, which are cells that are floating around in your blood, are more accessible to T cells. And for that reason, these types of strategies seem to work particularly well in them. So I can't tell you a specific example, but I would be willing to bet that TALL is being lined up and probably is already in clinical studies involving either BiTEs or CAR T cells. Very likely. Yeah.
Thank you for this superb presentation. So I have a close...
Thank you for that superb Boston accent.
I have a close relative who has pancreatic cancer, and the doctors are totally hopeless. Is it possible that some of the things that
Yes.
you discussed today
Yes.
could be focused on
Yes,
pancreatic
most
cancer?
definitely. Yes,
Really?
absolutely. So I should say that you guys have asked me about different cancer types, appropriately so. I think probably every cancer type is being pursued with one or another immune-based therapy. So in the case of pancreas cancer, I know of at least 3 things that are being tried. One is the CAR T cell type therapy. There are proteins that are expressed specifically on pancreas cancer cells, and T cells are being engineered to recognize those, and clinical trials are underway to test the efficacy of those treatments. So that's one thing. Pancreas cancer is tougher, however, because the cancer cells in the case of pancreas cancer are encased in what's called a stroma of non-cancer cells, almost a cement-like material that surrounds the cancer cells. And it looks like one has to break that down in order to more effectively allow the T cells to interact with the cancer cells, and there's work ongoing to do that too that I'm familiar with. So pancreas cancer, which is a disease we study in my lab, is one for which there's a lot of activity. We all recognize that it's a really hard challenge, but there's a lot of activity. So the answer is absolutely yes.
Thank you for the incredibly accessible lecture that you gave. My question is, you mentioned a preventative vaccine.
Yeah.
With just the sheer numbers of genetic mutations that you can have in a cancer, how do you select a target that is widely expressed
Yeah.
across many cancers and also that has just wide efficacy?
It's a good question. So, and there's a detail to your question that I'm going to kind of brush over, but so we emphasized the acquisition of mutations, and those are more or less patient-specific. They're not all patient-specific, but they're more or less patient-specific. Personalized preventative vaccines will never work in that setting, so you're absolutely right. However, things like HMG-A2 are commonly expressed. There's a whole suite of them, actually, across many cancers, right? So 50, 60, 70% of human lung cancers express HMG-A2, and likewise colon cancer, and by the way, pancreas cancer. So there are a number of these so-called shared antigens or tumor-associated antigens. And those are the ones that I'm more hopeful about, not the specific altered proteins. They have a different problem, by the way, a safety problem. And I didn't touch on that at all. We can talk about that over drinks if you wish. So it's not a panacea. We've got to solve the safety problem. But no, we would go after the shared antigens. Yeah.
All right. I think
Right, one more in the back. Yeah. Yeah.
Within the same patient,
Yes.
metastasis to primary tumors may be already very divergent.
Yes.
And that, like combinatorial treatments and immunotherapy.
Yes. It's a very good
can
question.
you repeat the question?
The question is that aren't you worried about tumor heterogeneity and tumor evolution? And it's a perfectly fair question. Tumors change over time. If they lose the expression of the thing you're targeting by your T cells or your NK cells or whatever cells, then they will become stealth to those cells. And so you're absolutely right. And probably some of the patients who are relapsing are suffering from that problem. The checkpoint therapies probably have less of that problem because they should have a whole suite of tumor-reactive T cells. So it's not focused on just one, but the CAR Ts could have that problem. So the hope is that in the setting of an effective immune response against a cancer, even if it's initiated by a single T cell, elicits a broader immune response in the same way that the immune system adapts to viral agents and bacterial agents. So you get what's called epitope spreading. So they don't focus on one antigen, and the immune system is rearming, in a sense, even as the tumor evolves. That's the hope. And the fact that those patients treated with checkpoint therapies do so well for so long makes me think that that's likely happening, actually. Yeah. Yeah.
So, one of the trooper pointed out that there was something like 400 clinical trials going on on combination therapy, and that there isn't a real scientific framework for what do you combine PD-1 blockade or CTLA-4 blockade with. What is that scientific framework that you would think that, how would you go about approaching that systematically, like for pancreatic cancer or bladder cancer?
The question is actually quoting a speaker that we had here in the Koch this week named Drew Pardall, who's an expert in this area, and he talked about the fact that there's 400 clinical trials looking at various combinations, and it's a little mindless, right? Pick one from column A and one from column B and see if they work better. And and he's right. We need better science to understand what's regulating the immune response in what patient in order to figure out how to better treat that patient. And so Anna mentioned the Moonshot Blue Ribbon panel, which I've been involved with. And there's a working group focused on immunotherapy. And one of their big questions is trying to distinguish the mechanisms of response and resistance patient by patient to be able to underlie or undergird those choices by real science. So we don't really know the answers yet. And Drew said the same thing. So we need more investigation. And I expect actually that the mouse models like we've created will help us figure those rules out. Yeah. All right. Thank you again for coming. I think there's drinks.
Desserts.